السلام عليكم، معكم دكتور أحمد جمعة. ودي انترو سريعة جدا لبرنامج يوجين for Bioinformatics. بالأخص ال Beginners in Bioinformatics. المرحلة الأولى اللي هو كيفية تنزيل البرنامج، طبعا هتروح لموقع eugene.net، تختار البرنامج اللي بيفت مع your operating system. Then you start it. You will find this is a welcoming page. You will find open files, create, run, or quick start. And all these tools, apps, with their files, actions, settings, and tools. You will find the tools. It's omics, all omics analysis and cloning, primers, design, and blasting for helping points. Now let's create a sequence. And in that case, we will go just in CPI, select any sequence from whatever locations you want, copy it, go back to UGENE and just paste the sequence in here and name your file and also select the location. Make the location desktop, it will be better for you. Create the file, and then you will have that overview for your sequence. The locus up. The sequence in details is just second section, and third section actually is the annotations and all the tasks that you're doing. You will find it below. Any task we will do in the up sections, it will be found in the down section. Now you will find the action menu has been activated. Here you can select any sequence, whether from above or down. These are two windows that are opening now, and all these tools you will find it actually in tool section and in analyze of action section. Okay, so let's start from the analyze, find pattern. Here we can just write in a small sequence, and that will show me the pattern if this sequence has been repeated or no. That is one algorithm for finding pattern, and you will find all the details for the advanced settings just below, as you see. Okay, now let's go back to the action menu and analyze, and we'll find a pattern with another algorithm. In that case, actually, he will select all the patterns in the whole sequence. In my case here, I selected and I found the annotation down actually. As you see, all these are the patterns for all the repeated sequences from the whole sequence I have. في الحالة دي I will go back again to the actions and I will find the ORF. You can select all the details and advanced settings for the ORF finding. Open reading frame. Here, all the ORF actually is a bit different from the online tools, which here is not numbered. So you can find the ORF just ORF. You can just select them from down here, whether finding the locations or just getting it copying and pasted. Let's copy another one and just paste it in Word file. It's exactly the same. So any ORF here, operating frame, it will be there. You can find it anywhere you want. And let's go back to the action menu and find the next tool. Annotates for the plasmid building tool. Now I just would like to find if is there any repeats in my sequence. Okay, this repeats actually can be a pattern for some kind of similarities between or relationship between organisms, or even sometimes actually within the genome itself it has some pattern. It means something. Like you will find here the finding pattern, finding the repeats, and in the other case actually you will find that the finding the tandem repeats. Here, the second one is the finding that tandem repeats. It's actually in this case, you will find all the tandem repeats within the whole sequence you have. You will find it down in the repeats, go down, 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 until okay, now is the restriction sites. If you would like to observe them, select whatever sequence, whatever restriction enzymes that you want. You will find all details about each enzyme actually, the cutting and even the suppliers. And I will search for this enzyme I checked in the whole sequence. That's why, okay, then you will find, just go down. You will find it here, the enzyme down in the auto annotation. Every single step that you're doing on UGENE, it's actually being recorded. So also you can design primers if you want. In that case, here's the primer C++. It's been included in that software in UGENE. And you can directly go to the primer from the annotations in the first part, the top primers. Here he gave you more than one pairs of primers. You will find the top and bottom primer, the forward and reverse one for each pair of primers. Okay, the M-fold actually, if I selected one of these primers and I want to check the dimers, if it has self dimers or no, like hairpin structure. So in that case, I will go to the M-fold. I'll just run it, select it region, and it will give me that it's already done. So I will just view the report. Here you can find this is structure, some kind of hairpin or self dimers. Secondary structure. Okay, this for most of the tools it's been used. So now let's go to the cloning part section. Also, you can here clone for plasmid or something. So in that case, actually, I have just digest my sequence based on my uh, restriction site that I selected before. And I have multiple fragments actually according to the restriction site I selected before. So in that case, I can create actually construct a molecule, and I can create fragment also as well. Here I will select one or two like fragments. In first case, it will be wrong a bit because I missed one thing. So I'll just repeat it one more time. Constructs, so adding it, and I will force that plant, and I will also make it circular. So now just click OK, and it will illustrate it for you. This is schematic representation about the fragments that you have connected together, okay, with all informations and annotations it has. Other than that, actually, you can repeat these processes more again and again until you become familiar with the GUI, the graphical user interface of UGENE. It's a very interesting one. Okay, now also I have inserted some sequences in my sequence, but I have first to activate the editing mode. Because of my sequence is a bit big and it has so many annotations, that's why it's giving me every time that's hanging my software. But it's too easy actually to insert sequences once you go from the actions and you edit and you switch on the editing mode. In the next sessions, I will go through the omics in more details regards to the NGS and also Sanger and other algorithms for assembly, genome assembly, and next generation sequencing and all these tools. Thank you so much.